So welcome you all. So here we are going to discuss solved question paper of Biochemistry paper 1, and the paper is by from MUHS University. So here we are going to discuss brief answer question, any 10 out of 11, and these are the questions. So we will discuss all the questions one by one. These are the solved questions. So the first question is, name two diseases due to the altered lysosomal function. So here, due to the altered lysosomal function, what are the diseases? So here the answer is GM2 gangliosidosis. So in which there is a type A, type O, and type AB. Type A, that is a Tay-Sachs disease. Type O, that is Sandroff disease. And type AB, that is a GM2 activator deficiency. So here just only remember the name. You can just write Tay-Sachs disease, Sandroff disease. Here you can write Niemann-Pick disease, Gaucher disease, Fabry disease, and metachromatic leukodystrophy. They have only asked write any two diseases. Just write Tay-Sachs, Sandroff, Niemann-Pick. Any two from this you can write. That will be the answer for altered lysosomal function. Next one is give the chemical composition of hyaluronic acid and chondroitin sulfate. They have asked only the chemical composition. So for hyaluronic acid, the chemical composition is D-glucuronic acid and N-acetyl D-glucosamine. For chondroitin sulfate, the composition is D-glucuronic acid and N-acetyl D-galactosamine 4-sulfate. So this was asked for 2 mark. For future, they may ask what is the composition of heparin, that is a L-iduronic acid 2-sulfate and D-glucosamine 4-sulfate. The composition of keratin sulfate, that is a beta D-galactose and N-acetyl D-glucosamine 6-sulfate. Dermatan sulfate, the composition is L-iduronic acid and N-acetyl D-galactosamine 4-sulfate. Okay. Then the next question was define liposomes and state its uses. So liposomes are spear shaped. These are the spear shaped vesicles consisting of one or more phospholipid bilayer. यहाँ पे it is a spherical or it is spear shaped vesicle है, and which is consist of one or more phospholipid bilayer. यहाँ पे one or more phospholipid bilayer है, and it is prepared by sonication. These liposomes, which are commonly used for the delivery of drugs, delivery of proteins and enzymes. So these drugs, proteins and enzymes, these are the may be encapsulated by liposome, which act as a carrier for substances to target organ. So here it is commonly used for the treatment of cancer. Cancer के लिए which is it is commonly used, where you can see this is a central core in which drug is kept and it is transferred to the target organ. It can be used as an antimicrobial, it can be used for the gene therapy, and it is used for the diagnostic imaging. Then next question was give reason why excessive ingestion of alcohol results in hypoglycemia. So alcohol, that is your ethanol. This ethanol which utilizes nicotinamide adenine dinucleotide. So this NAD which get convert into NADH and ethanol get convert into acetaldehyde. So during alcohol consumption, excessive alcohol ingestion, what happens? There is no glucose is there, so there is a process called as a gluconeogenesis occur because of unavailability of glucose. There is a gluconeogenesis, and the substrate for that is a pyruvate and oxaloacetate. So for the gluconeogenesis, we need NAD. हमें यहाँ पे ग्लूकोनियोजेनेसिस के लिए NAD चाहिए होता है, but excess of this NAD which is utilized for the this oxidation of इथेनॉल टू एसिटाल्डिहाइड, जो भी NAD यहाँ पे था वो NAD गेट कन्वर्टेड टू NADH। यहाँ पे NAD का कन्वर्शन हो गया है NADH। So during gluconeogenesis, there is the unavailability of NAD. That's why the gluconeogenesis that is hampered, and the conversion of pyruvate and oxaloacetate into the glucose by the process of gluconeogenesis is impaired. Due to that, due to alcohol consumption, gluconeogenesis is reduced, which result into hypoglycemia. Okay, so यहाँ पे ग्लूकोज नहीं बनेगा from the non-carbohydrate precursor, that is a pyruvate and oxaloacetate. Then next question was name four inhibitors of complex 2 of electron transport chain. So the inhibitors of complex 2 are carboxyl and TTFA, which specifically inhibit transfer of reducing equivalent from succinate dehydrogenase to FAD, and malonate, it is a competitive inhibitor of succinate dehydrogenase. So always remember these are the inhibitors of complex 2 of electron transport chain: carboxyl, TTFA and malonate. Then next question was name any two preventive antioxidants. So always keep it in mind कि preventive antioxidants क्या होता है. These preventive antioxidants, they inhibit initial production of free radicals. They prevents or they inhibit the production of free radicals. The examples are catalase and glutathione peroxidase. ये है preventive antioxidants. The examples are catalase and glutathione peroxidase. Then may be another question that what are the chain breaking antioxidants? कौन से chain breaking antioxidants है? This what is the meaning of chain breaking antioxidants? They inhibit the propagative phase. ये propagative phase को inhibit करते हैं. And which are they? The examples are superoxide dismutase and vitamin E. The answer is specific. The answer should be specific. If the question is asked what are the chain breaking antioxidants, the answer should be superoxide dismutase and vitamin E. If the question is preventive antioxidants, the answer will be catalase and glutathione peroxidase. If the question is antioxidant used as a therapeutic agent, so answer will be vitamin E, vitamin C, dimethylthiourea, dimethyl sulfoxide, and allopurinol. These are the therapeutic antioxidants. These are the remaining questions from BAQ. Now the next question is enumerate any four factors affecting nitrogen balance. So what is the meaning of nitrogen balance? Nitrogen balance, that is the difference between intake of nitrogen and excretion of nitrogen. So in which form we are taking nitrogen? That is nothing but a protein. Protein के form में हम nitrogen लेते हैं, and the excretion of nitrogen is that what the difference between intake of nitrogen and excretion of nitrogen. Whatever the balance is there, that balance is a nitrogen balance. So what are the factors affecting nitrogen balance? So there are two types. One is positive nitrogen balance and second one is a negative nitrogen balance. What is the meaning of positive nitrogen balance? It means that nitrogen, it is stored in a human body. वो कब store होगा? During infancy, during childhood, during adolescent phase, during pregnancy and growth, during recovery from severe illnesses. During pregnancy, what happens? There is a fetus, intrauterine growth of baby that need nitrogen. So nitrogen, it is stored in a baby. So that is a positive nitrogen balance. Then growth during recovery from severe illnesses. अगर patient chronically ill है and in that condition commonly we give egg protein or amino acid supplementation for the proper growth and development or growth of that patient. So During that growing phase, the nitrogen is stored and there is a positive nitrogen balance during recovery from severe illnesses. Then next one is the negative nitrogen balance. Negative nitrogen balance means loss of nitrogen, and this thing commonly happen during trauma, during cancer, during uncontrolled diabetes mellitus, and severe nutrition deficiency. So all this catalytic activity, all the protein, there is a breakdown, and whenever there is a severe nutrition deficiency, in all this condition there is a negative nitrogen balance. Then next question was enumerate any four enzymes of diagnostic importance in a liver diseases. So you have to just write only the four enzymes for liver diseases. The examples are SGPT, that is serum glutamate pyruvate transaminase, and the range is 10 to 35 international unit per liter. Then SGOT, that is serum glutamate oxaloacetate transaminase, and the range is 8 to 12 international unit per liter. Next one is the alkaline phosphatase, that is the 3 to 23 King Armstrong unit per deciliter. Then gamma glutamyl transpeptidase, the range is 5 to 15 international unit per liter, and 5 prime nucleotidase, that is the range is 2 to 15 international unit per liter. The normal ratio of HGPT to HGOT or AST to ALT ratio is 0.8. If ratio is more than 2, it indicates that patient is suffering from myocardial infarction, patient is suffering from alcoholic hepatitis, and patient is suffering from cirrhosis. So the question may be asked, in which condition the ratio is more than 2, in which condition the ratio is decreased. If the ratio is less than 0.8, it indicates that patient is suffering from hepatocellular injury or cholestasis. Then next question was give fasting and postprandial blood sugar level in impaired glucose tolerance or it is a pre-diabetic condition. So what is the meaning of impaired? Whenever the abnormal glucose homeostasis, it is defined as a fasting blood sugar level between 100 to 125 milligram per deciliter, which is defined as a impaired fasting glucose. And here the answer is the fasting blood glucose level that is between 100 to 125 milligram per deciliter. It is a pre-diabetic condition. Similarly, the postprandial blood glucose level in impaired glucose tolerance is 140 to 199 milligram per deciliter. The answer was fasting and postprandial. This altered value indicates impaired glucose tolerance. The next question may be write the ADA criteria for diabetes mellitus. So the fasting value for this American Diabetic Association, the fasting value of blood glucose level more than 126 milligram per deciliter, that is the fasting one, and the postprandial blood glucose value more than 200 milligram per deciliter, along with glycated hemoglobin more than 6.5%. It indicates that patient is suffering from diabetes mellitus. That is the criteria for confirming diabetes mellitus according to ADA, that is American Diabetic Association. Then next question was enumerate two oncosuppressor gene. So these are the gene, that is RB gene, that is the oncosuppressor gene, and if it is converted, activated, there is a retinoblastoma is there, P53 gene, VHL gene, APC gene, BRCA2 gene, NF1 gene, and PTCH gene. So all these genes are oncosuppressor gene. You have to answer, enumerate, the question was asked, mention any two oncosuppressor gene. Okay. Next question was enumerate any four functions of cholesterol. As we know that cholesterol, it is a very important, it is commonly asked question for Viva, and the four functions are asked. So it is a constituent of the cell membrane. It is an important constituent of cell membrane. This cholesterol is also utilized for the synthesis of bile salts. The specialized compound formed from cholesterol are vitamin D, steroid hormones like cortisol, estrogen, and testosterone. So these are the specialized products which are formed from cholesterol, and vitamin D is also an important product formed from cholesterol. Then now SAQs, short answer questions. Any 8 out of 9. So this is the first case in that range. The question was, a baby showed the presence of congenital cataract and liver enlargement. The blood investigation shows elevated galactose. There are also presence of galactose in urine. So it is a clear-cut case of galactosemia or classic galactosemia. So here there is a hepatomegaly is there and cataract is there. The catching point is galactose elevated है blood में, galactose present है urine में, cataract है और liver enlargement है. It means that the disorder is galactosemia. The question was name the probable disorder. The answer is classic galactosemia. Next question was mention the deficient enzyme. So the answer is galactose 1 phosphate uridyl transferase. This enzyme is deficient, and here there is accumulation of both galactose and galactose 1 phosphate in the blood and in tissues like RB cells, liver, the RBCs, liver, spleen, kidney, heart, lens of eye, and cerebral cortex. So here this accumulation responsible for the side this manifestation. So accumulation of galactose 1 phosphate responsible for hepatomegaly, and in lens there is a formation of galactitol or dulcitol. So that is responsible for cataract. The another enzyme deficient is galactokinase. Galactokinase deficiency, it is a minor type, and in these galactose accumulate into blood and tissues. So this classical galactosemia, it is the most common type where there is a deficiency of enzyme galactose 1 phosphate uridyl transferase. So in management, you have to avoid galactose in a diet. The next question was why treatment is not required beyond 4 year of life. 4 साल के बाद हम इसमें हमें galactose free diet देने की जरूरत क्यों नहीं पड़ती है? That is a special diet regimen that is recommended for the management of galactosemia. So it is a withdrawn after 4 years. 4 साल के बाद हम उसको withdraw कर देते हैं. Why? Because here the galactose 1 phosphate pyrophosphorylase becomes active. भले ही यहाँ पे galactose 1 phosphate uridyl transferase deficient है, लेकिन 4 साल के बाद galactose 1 phosphate pyrophosphorylase नाम का enzyme activate हो जाता है, and that will take care of that disorder. So galactose 1 phosphate pyrophosphorylase becomes active, and that will take care of galactose, and the features like cataract and liver enlargement that will not develop after the age of 4 years. That's why it is recommended that special diets that is withdrawn after 4 years. Next question was explain the biochemical basis of the development of cataract. हम यहाँ पे cataract होता है तो क्यों होता है? The reason is that excess of galactose in a lens is reduced to galactitol. Galactose है जो लेंस में जमा हो गया था, that get reduced to galactitol और dulcitol by the enzyme aldose reductase. Aldose reductase नाम का एंजाइम जो लेंस में रहता है वो गैलेक्टोज को गैलेक्टिटॉल में कन्वर्ट कर देता है. Galactitol, that is once it is trapped into the lens, that cannot escape from the lens, and it has an osmotic effect. The sugar जो है वहाँ पे galactitol है और sugar alcohol है, that contribute to the injury of lens protein, और वहाँ पे there is a development of cataract. It has tendency to hold water and responsible for damaging lens proteins and development of cataract. Okay. Again, the thing is that excess of galactose inhibit glucose 6-phosphate dehydrogenase. As we have seen that is glucose 6-phosphate dehydrogenase that is in HMP shunt, that is very important for the formation of NADPH. And this NADPH, if it is not formed, that may be result into the damage to the lens protein by the free radical, or which result into the reduce less production of reduced glutathione. As we know that this reduced glutathione that take care of फ्री रेडिकल, अब यहाँ पे एनएडीपीएच नहीं बनेगा, तो वहाँ पे रिड्यूस जीएसएच भी नहीं बनेगा, और जो फ्री रेडिकल है वहाँ पे दे कैन इजीली अटैक लेंस प्रोटीन that may damage lens protein and responsible for development of cataract. So these are the two mechanism or two biochemical basis for the development of cataract. So in this lecture, in this video, we will discuss only BAQ and one SAQ. This is part one. In part two, we will discuss remaining questions. Okay.